Bismillahirrahmanirrahim. Today I would like to share the knowledge about protein purification. My name is Sarfraz Rahman. I did PhD in parasitology. I would like to share this knowledge with references this book, Cloning and DNA Analysis, 6th edition by T.A. Brown, and internet sources. Please subscribe my channel and also push the button of bell icon. So first of all, you should know about what is the protein purification. It is a series of processes intended to isolate one or few proteins from a complex mixture, usually cells, tissues, or whole organisms, vital for characterization of function, structure, and interactions of the protein of interest, separate the protein and non-protein parts of the mixture, and finally separate the desired protein from all other parts. So the separation of one protein from all other is typically the most laborious aspect of the protein purification. So the goals of protein purification: obtain a particular protein, especially the recombinant protein, from others and their cell components, and proteins are used throughout biological and biomedical sciences. Obtain a good yield, absolute amount and proportion of starting amount, and maintain the activity of the protein. So the protein purification factors, the protein purification method use for fraction techniques, which are in a large part based on solubility, size, charge, binding specificity. So the methods for protein purification: the different methods are used for protein purification: crude extract, centrifugation, dialysis, column chromatography, gel filtration and size exclusion, mass spectrometry. So protein purification by the nickel NTA column, its basic principle is that the nickel NTA column protein purification system is based on the remarkable selectivity of the patented nickel NTA nickel nitrile triacetic acid resin for protein, especially the recombinant protein, contain an affinity tag of 6 or mix more histidine residues consecutively or alternating the his tag. So this is the brief method how to do the purification of the protein. First of all, you need the 50 ml culture, which is induced with the help of IPTG, and then you did centrifugation 5000 G for 5 minutes. Discard the supernatant, then resuspend the pellet and with 2 ml lysis buffer, then sonication, and then after the sonication you did the centrifugation 8000 G for 30 minutes at 4 degrees centigrade. Then you get the two tubes, one is the cell lysate and add the 2 ml extraction buffer, other is the cell pellet and add the 2 ml of the extraction buffer. This is the method according to this kit, that is the catalog number is the 88228, that is the Thermo Fisher kit. So protein purification by the nickel NTA column And the first step is that the equilibrate the column by adding the 2 ml equilibrate buffer. This 2 ml equilibrate buffer means that it is equal to that the resin bed volume. And then centrifugation at 700 G for 2 minutes at room temperature, and repeat this step of this equilibration. Then add the protein extract into the column. Put the column at rocker for 60 minutes. Centrifugation 700 G for 2 minutes. Add wash buffer 2 ml into the column. Centrifugation 700 G for 2 minutes and repeat the process. And add illusion buffer 2 ml into the column and repeat for 3 times. And after that you should observe this quantity of the protein with the help of the spectrophotometer at 280 nanometer. So this is the method how to prepare the nickel NTA column, and these are the components which is needed to prepare the nickel NTA column: the nickel chelating resin and nickel chelating spin column, nickel sulfate hexahydrate, and MCAC metal chelate affinity chromatography, and it has the following component: sterile glass wool and column stand which is made up of the iron. So next this is the brief sketch of this procedure. First of all, you put the glass wool in the column, add 0.2 ml NTA resin slurry to 1 multiple 10 centimeter, which is the 5 ml volume of the the column. Wash the column with 1 ml 5 bed volume deionized water. Charge the column by washing with 1 ml 5 bed volumes of 100 millimolar nickel sulfate hexahydrate. Wash the column with 2 ml metal chelating affinity chromatography buffer. When the charged with nickel ion, it has a light blue-green color, and when stripped of the nickel, it is white. And see the instruction of the resin preparation. And charged resin can be stored at 4 degrees centigrade. If the column is to be stored more than 1 day, washed with the 10 bed volume of the 20% of this alcohol. So this is the process. This is the brief procedure of how can you prepare the nickel NTA column. And this is, if you have any query, you can ask the question. And thank you so much.